So there's a lot of current research going on about induced neurons. One study I found was from the University of California, and it showed that it's possible to reprogram astrocytes into neurons. Another study I found was from the Karolinska Institute in Sweden, and it used neurons to treat Parkinson's disease by reprogramming fibroblasts into dopaminergic neurons in rats. And a third study I found was from Penn State University, which injected a cocktail of transcription factors into the brain of mice after a stroke, and it significantly improved recovery. There are a few limitations with Lieb's study. One is limited conversion efficiency. Most of the converted astrocytes into neurons were observed in the hippocampus and cerebellar cortex, and not many in the striatum or cerebral cortex. Another limitation is localization issues. Most of the conversion was found where the injection was, suggesting that effective reprogramming of astrocytes to neurons is highly localized and possibly influenced by the injection itself. Another limitation is the viral vector challenges. This is difficulties achieving specific targeting of astrocytes, and this led to low expression levels and off-target effects. And the last limitation is the promoter activity persistence. In cells that underwent conversion, the expression of the HGFAP promoter remained active, which is atypical for mature neurons. This means that some of these neurons may have not fully matured and may maintain some characteristics of astrocytes. There are many potential applications of induced neurons. One is disease modeling. Induced neurons can be used to create specific conditions of certain diseases, which will allow us to study them better. Another application is drug discovery and testing. While being able to create neurons can allow us to more easily test drugs and their effects on these neurons. Another is personalized medicine. Induced neurons can be generated from a patient's own cells, allowing them personalized medical approaches. Another is regenerative medicine. Certain neurodegenerative diseases or spinal cord injuries or stroke may have damage to neurons, which at the current moment the body is unable to regain. But using induced neurons, we can help patients get some of these functions back. Another is neural interface development. Research in creating interfaces between electronic devices and the nervous system can benefit from induced neurons, such as Neuralink. In understanding neuronal development and function, induced neurons help scientists understand the process that is involved in neuronal development. So what is the significance of induced neurons? Well, like I mentioned before, they help us understand neurological diseases. So diseases like Alzheimer's, Parkinson's, and Huntington's disease, which are neurodegenerative diseases. Induced neurons can help us understand how these neurodegenerative diseases work and how we can help fix them and understand the underlying mechanism of what causes these. Next is drug development and screening. Using induced neurons derived from patients with specific disorders enables high screening of potential drug treatments. It allows for the testing of pharmacological responses in a patient-specific context, specifically improving the process of drug discovery. And lastly is regenerative medicine. Induced neurons hold potential in regenerative medicine to treat injuries or diseases that result in neuronal damage or loss, such as spinal cord injuries, stroke, or neurodegeneration.